All right. Hi, everybody. I'm going to give a couple general announcements. One, you should have received an email from me a little bit before this with a link to a doodle poll for a review session because the next exam will be next Friday. All of the old exams and things are already posted. Please reply to the doodle poll by Monday at class time so that I can tell you in class on Monday when the review session will be. And feel free to send me emails, come by office hours, all those kind of things if you want to talk about the old exams and practice questions. And remember that the T-cell development problem set is due today by 5. So today we are going to talk about T-cell activation. And basically, as I mentioned to you before, it's basically sort of the beginning of going through the events that are shown on this slide. And this is largely going to be what we're going to be talking about from now until the exam, are the events that you see here. Where we see that, if you recall last time, we got our naive T cell into the lymph node in the same place where we had our dendritic cell potentially presenting antigen so that the two can interact and so that the T cell can undergo some of these steps of activation to eventually become an activated T cell that's going to do something about our microbe. And as I told you last time, really today we are zooming in here at the interaction between the antigen presenting cell that is presenting on MHC along with the T cell receptor. So we're kind of zooming in on details that are happening right here, where we have the MHC molecule plus peptide binding to the T cell receptor. Remember, the T cell receptor needed some partner proteins, including CD3, which was important because CD3 has some long cytoplasmic tails that provide ITAMs, which are tyrosines that get phosphorylated. And recall that we have either CD4, if we are talking about a CD4 T cell interacting with MHC class 2, or CD8 if we're talking about a CD8 T cell interacting with MHC class 1. And either of those has a kinase that is associated with its cytoplasmic tail. So when MHC plus peptide comes close to this T cell, we're going to pull our CD4 or CD8 next to the T cell receptor with CD3. That's going to pull this kinase in close proximity to these ITAMs, and now the kinase is going to be able to phosphorylate the ITAMs. So we kind of saw this before. As I told you when we talked about this before, one of the reasons why that CD4 that's shown here or the CD8 that would be on a CD8 T cell is super important is that the CD4 or CD8 is bringing in this partner kinase in order to do the phosphorylation and start signaling. I also told you that this CD4 or CD8 is important because the binding affinity for the T cell receptor to MHC plus peptide isn't super strong. And so if we want these two cells held together for enough time to kind of really let this signaling process happen, we need additional partners. CD4 or CD8 is one of those additional partners in that it is also making contact with the MHC molecule. If we are being realistic, there are way more proteins on the surface of the T cell and on the surface of the antigen presenting cell that are actually holding these two cells together. So it's not just the proteins I showed you on the previous slide, there actually are a huge number of proteins that are interacting between these two cells and holding them together. And so you can see an example of many proteins on the surface of the T cell interacting with many proteins on the surface of the antigen presenting cell. This shows this for both a CD4 and for a CD8. One thing that might be of interest to you is if I'm not going to go through the whole list of all the proteins that are holding these two cells together. I'm only going to mention sort of one other protein outside of the T cell receptor, CD4 with MHC plus peptide, in terms of like really specific information. But if I were, and part of the reason I'm not going to go through all the specifics is because it's different in different situations. But one of the things that actually is happening here, if we actually were to make a list of all of the proteins that are holding them together, is if you look on this image, you see LFA and ICAM1 here, LFA and another binding partner, CD2 here. Those are actually integrins, and you saw integrins last time that have that super strong binding affinity. And so we actually have some integrins that are really tightly holding these two cells together. So we do have other interactions that are kind of making sure these cells are held together tightly. I'm not going to go into tons of detail about this. I used to, and the field has sort of changed a little bit in the way that we talk about it, so sort of don't feel like it makes sense to go as deeply into right now. But I want you to think for a second about those integrins. You can see that those integrins are shown here on kind of the outside, and a lot of the other proteins that are interacting are sort of inside of them. If you actually look at an image of this, it kind of looks like the Target logo, that the integrins are like making a circle around the outside and all the other proteins are making a little circle on the inside. And that actually is kind of important because it like focuses all of the interactions between the cells, sort of between the integrins. The integrins are almost like a little wall. What's going to happen also later on is we're going to see stuff get secreted from one cell to another. And that secretion actually happens between this integrin wall. So it's like if the T cell secretes something that it wants to go to the antigen presenting cell, or the antigen presenting cell secretes something it wants to go to the T cell, the stuff can't like diffuse out randomly anywhere in the cells. It has to stay inside that integrin wall. So I point this out because we talked about integrins last time, and their high binding affinity. This is another place where integrins become important. When we are talking about the activation of T cells, this is the big picture part of the story. I'm not going to go into all the parts of this story today, but I will tell you this is sort of the spoiler of where we're going to end up. In that when we have a naive T cell, when we have a T cell that is getting activated for the first time, it needs to receive multiple kinds of signals. There is the signal that comes from the T cell receptor, which you can see here, that is often referred to as signal 1. And so I'm going to talk about signal 1 a lot. You can also see that there is this additional signal that has to happen called signal 2 that involves some other types of proteins. And further, you can also see that there have to be some cytokines involved in this process. Some people call the cytokine signal 3. If we call it signal 3, I don't care. But really, if our T cell is a naive T cell, if it is completely fresh out of the thymus, it needs signal 1, signal 2, and signal 3. When we are talking about this stuff today, we are largely, and so you can see here in this image from your textbook, sort of the events of signal 1, it has a 1 at the top, coming from the T cell receptor, and you can see events of signal 2 happening from some other proteins. By and large, today, I'm only talking about events that are considered part of signal 1. So today is really going to be a very signal 1 kind of day. I might, at the end, depending on how far I get, actually talk a little bit about signal 3. Signal 2 is happening next week. But signal 2 is like so crazy important. I just want you to already know, like, yeah, it's really important, but it's separate from the stuff we're talking about today. And so really, the main set of things I want to tell you about today are the events that happen in the T cell downstream of the T cell receptor. So when the T cell has its T cell receptor, CMHC plus peptide, what events happen in the T cell to take that T cell from a naive T cell to an activated T cell? What actually happens inside that T cell? I've already told you a little bit about what happens, and you're going to see that on some later slides as well. So you'll see, I'm going to show you slides that basically tell you things I already told you verbally today and verbally in the past. But this is sort of your textbook's overview of all of this. You can see that we have our T cell receptor, and there are a number of different types of signal transduction proteins that are involved in signaling in that T cell, and that eventually lead to changes in transcription because of transcription factors. So we got a bunch of different steps of this. You can actually see that in this image on the left, where you can see the T cell receptor and you can see the B cell receptor. And honestly, the themes are identical. So this image is showing you it's basically the same identical big picture steps. The little picture details of like which kinase differ in certain situations, but the fact that there is a kinase from this group and then there is a protein from this group is the same. And your textbook kind of shows us largely what we're going to be talking about today in terms of what happens with the T cell receptor in this one figure, figure 8.11. So it's kind of like today is going to be largely about figure 8.11, but we're actually going to like go through it in detail and not just have to look at some random words, and really talk about what is being described here. But there's one key thing I want you to see about figure 8.11 that is going to be really critical for what we're talking about today. So as we talk about this process of activating a T cell, there are tons of things that we could say are like what happens to a T cell when it's activated, the end goal of T cell activation, what a T cell does when it's activated. And so for us, I want to tell you what goal we're talking about. So figure 8.11 mentions this here. And figure 8.11 mentions 3 key transcription factors. And so our goal in activating the T cell through the signaling we're going to see today is turning on those 3 transcription factors. One of them is called NF-kappa B. For some reason, everyone wants to call it NF-kappa beta. It's actually NF-kappa B because it was originally described looking at the kappa chain in B cells. And the NF is nuclear factor, which is an old-fashioned term for transcription factor. So it's the nuclear factor that was important for the kappa chain in B cells. One called NFAT, which is the nuclear factor in activated T cells. And one that's called AP1. And so our goal today is getting active, getting ready to go versions of NF-kappa B, NFAT, and AP1. And you can see this here as well. Our goal is we want to get NFAT NF-kappa B, AP1. Yes, they are written in a different order at the top of this slide, but it's the same 3 transcription factors, and there is actually no particular order for them. And one of the reasons why the combination of those 3 transcription factors is going to be so important for us is that if a T cell has all 3 of those transcription factors active and ready to go, that T cell has the ability to transcribe a particular gene. And that gene is the gene for IL-2, so it encodes the protein IL-2, which is a really important cytokine in T cell biology. So our goal in all of the signaling that we talk about is getting NFAT, AP1, NF-kappa B. And the reason why we care about those 3 is because they give us IL-2. So as we're talking about all of the sort of minutiae of signaling, know that it's always about NFAT, AP1, and NF-kappa B because we're trying to get to IL-2. When we talk about signal transduction, one way that people sometimes divide up signal transduction is they talk about signal transduction in terms of something called receptor proximal signaling, which means like the beginning stuff, the stuff that's right next to the receptor, or later stuff. And so technically, what I'm showing you in the next couple slides are receptor proximal events. They're the ones that happen right next to the receptor. They're the first things that happen after the receptor gets stimulated. And we'll see other ones later. In some ways, I call it receptor proximal signaling to like remind myself for organization of what I'm talking about. But there's another reason why it is kind of important for you to think about whether or not we're thinking about something that's receptor proximal or whether we're thinking about something that's further down in the pathway later. And so here you see our receptor proximal signaling. Originally, the T cell receptor is hanging out with CD3, so they are always hanging out together because of interesting interactions in their transmembrane domains. CD4 is diffusing separately in the membrane, so they're usually like some different spot around the surface of the cell. This drawing is trying to show them far apart. That's not very far, but the drawing's trying to show us far apart. And if MHC plus peptide comes around, now that MHC plus peptide is going to bind to the T cell receptor. It's also going to bind to CD4 or CD8. And so these 2 things that were originally far apart are going to get pulled close together. Their extracellular domains are getting pulled close together. Here you can see how close together they are pulled. I wouldn't draw this exactly like this, but I also can't draw, so who am I to judge? When those extracellular domains get pulled close together, the intracellular domains are also close together. And so now we have this kinase that likes to hang out with CD4 or CD8 that's getting pulled really close to all of these ITAMs that are shown as sort of little yellow bars. This image shows the kinase as being a kinase called LCK. And you can see LCK in both versions of this slide. Because I like details a little too much, sometimes it makes me feel a little twitchy when it says it's LCK. Only because it's actually only LCK in the case of a CD4 T cell. If it's a CD8 T cell, it's a different kinase called FYN, F-Y-N. If you tell me that it's LCK in a CD8 T cell, I'm not going to get mad at you. The thing that's important with either LCK or FYN, F-Y-N, is they're both super closely related kinases. They're both in the same kinase family. So those of you who may be taking molecular biology of cancer with Dr. Dunaway, we've got LCK, we've got FYN. They're both members of the Sarc family. So these are both Sarc family kinases that you may have seen in molecular biology of cancer. So we've got our Sarc family kinase shown on this slide as LCK getting brought into proximity of these ITAMs, and now LCK is able to add a phosphate group to all of these ITAMs. And you can see that indicated by the pink dots added to the ITAMs. A kinase is an enzyme that phosphorylates things. This kinase specifically phosphorylates tyrosines. ITAMs are full of tyrosines, so we get phosphorylation on all of our ITAMs. And so you can see that here. And then we should think a little bit about what sort of happens with our phosphorylated ITAMs. So when we think about anything getting phosphorylated, particularly a tyrosine like we see here, we end up seeing a change in charge happening. We also see basically a change in kind of biomolecular shape happening. The way you can think about this change is, again, if you want to think about this as just there's now a new shape that wasn't there before. See, now there's like a rectangle with a pink dot on it. There wasn't before. New shape. Like I told you, I think once before, when I was in college, I told one of my non-science friends that immunology was just about matching shapes. It was actually preschool. And just do the shapes match or not. So we got a new shape. So now we have a site where things can bind that wasn't present before. Because there's this new shape, so stuff could bind that couldn't bind before when that shape wasn't present. So now that we have this new shape, we can bring in a new partner who has the ability to bind to this shape. This partner couldn't be here before because its binding site was not present. And so once we have our phosphorylated ITAMs, this brings in the next member of the process, which is a protein called ZAP70. And so you can see ZAP70 here. You can also see ZAP70 here in the slide that I was supposed to go to as the next slide. I don't know why I skipped ahead. So here you can see we get these phosphorylation sites, and that lets ZAP70 come in. Here's your textbook's version of it. You can see ZAP70 has this little shape on it that's really good at binding phosphorylated ITAMs. Before, ZAP70 can't really do anything because there's no phosphorylated ITAMs for it to bind. Now, once we have phosphorylated ITAMs, ZAP70 can also come in and start binding in this place where it could not before. So we've now brought in this new partner. Yep, Paige.
Is ZAP70 like around there, or is it like the CD4? Like somewhere
It's somewhere else, honestly. Their drawing of somewhere else is still kind of close. But yeah, so it's somewhere else, and it gets brought in because it now combined to this new shape that wasn't present before. Great question. So ZAP70 has enzymatic activity. So it is an enzyme, it can do something. And so before, we didn't have this ability in this area. Now we've basically brought in ZAP70's enzymatic activity to this location. ZAP70's activity is that it is also a kinase. So ZAP70 is also a kinase. It's going to phosphorylate stuff. You don't necessarily need to get super hung up on what it's phosphorylating. If I'm being perfectly technical, LCK doesn't get every single one of these ITAMs. ZAP70 actually finishes the job. We don't have to get that stressed. Just know this kinase, ZAP70, comes in, it phosphorylates stuff. In fact, it itself even gets phosphorylated. So we got this kinase that comes in. Once we have phosphorylated ZAP70, we go to the next step of this process. And in the next step of this process, a few things start to change. So once we have phosphorylated ZAP70, that provides binding sites for 2 more partners. So once ZAP70 is here, we get the next 2 partners. Their names are LAT and SLP76. I'm basically going to just say LAT and SLP76. I'm going to say them interchangeably. I'm not going to say anything that's different about them. I'm just going to sort of say there's 2 here, which is true. There's LAT and SLP76. So once we've got ZAP70, we're going to active. That's going to allow us to bring in LAT and SLP76. And LAT and SLP76 are a different kind of protein than we have seen elsewhere in this pathway. So they have a different role than what we've seen for other proteins in this pathway. LAT and SLP76 are what are called adapter proteins. And so after ZAP70, we bring in our adapter proteins. When you hear about adapters, when you hear the word adapter protein, one thing you might think about is what might happen if you travel to another country. So I go to South Africa from time to time. And when I go to South Africa, I have to bring an adapter with me to South Africa. What is this adapter that I bring with me to South Africa? You can imagine this in traveling to lots of other countries. It's not like a South Africa specific thing. Yeah, Jazz?
Within the context of an adapter itself, it's almost like a bridge between your device and the outlet.
Yeah, so in South Africa, the plugs look different. And so if I want to charge my phone or I want to charge my laptop, I can bring my cord and it won't plug into the wall, right? So I have this little device that's a bridge, exactly as Jazz said, between my plug from my laptop and the wall in South Africa. So I can charge my laptop. Jazz, does that little device, that bridge device, that adapter that I have, does it actually do anything? No, it has no function itself. It's just serving to be a bridge between 2 different things. In fact, that's actually a pretty important thing for me to know. The electricity type in South Africa, there's actually some differences in the electricity in South Africa. And some of my devices can't handle it. And the adapter is not like switching up the electricity for me. So if I bring my hairdryer, my very fancy hairdryer that I have, it will kill my hairdryer. So I can't bring it. Because in fact, the adapter is doing literally nothing other than being a bridge. These adapter proteins, you can think of in exactly the same way. They have no enzymatic activity whatsoever. Their job is to be a bridge from the signaling we see here in the receptor proximal signaling, the signaling we see with ZAP70, the signaling we see with LCK, and the signaling we're going to see later. Basically, they have no enzymatic activity. They're doing nothing other than bridging 2 things together. And so you can see this happening here. Basically here, this image is calling it a scaffold, but it's the same thing. It's an adapter protein, and it's this green pill-looking thing. It can get phosphorylated, and as a result, it can be the binding site where a ton of things can bind and get brought together that normally wouldn't get brought together. So normally, like orange protein and yellow protein would never interact. But since they can both be bridged by this adapter, suddenly they're brought close together and can interact. And so basically the adapter is just like this place where a bunch of other proteins can come in and interact. And so you can see in our signaling cascade at the top, here's LAT, here's SLP76, and they are just, they actually get phosphorylated by ZAP70, and that just gives sites, gives new shapes, so a whole bunch of other stuff can bind. So it's basically this thing that's just like sitting there to be a bridge and to let other stuff come in and get bound to it. And as you can see in either of these images, our adapter protein is bringing in a lot of stuff. We're getting a lot of things binding, and you can see that happening here in all of these cases. And those things that are bound, those things that come in, can sort of lead to different effects. And so once we get to the adapter protein part of the signaling cascade kind of branches into different directions. Before the adapter, it's all sort of the same. Once we get to the branches, or once we get to the adapters, we start to get onto different branches. And I will come back to that a little bit later about why kind of top where there's no branches and bottom where there are branches is important. And so after our adapter proteins, I have to then tell you about branches. And so what we're going to do is we've seen LCK function, then we've seen ZAP70, then we've seen our adapters LAT and SLP76. Now we're going to have branching happen. And so I'm going to go down one branch all the way till we get to a transcription factor. And then after we get to that transcription factor, I'm going to come back to LAT SLP76, and I'm going to go down again until we get to a transcription factor. So that's what kind of the way that we're doing all of this. So the first thing we need to do is we need to think about the first branch. And I'm not even going to go to the slide of the first branch. We're going to talk about it, and then I'm going to show you the slide. Okay? Because I'm going to ask you some hypothetical questions. So way back in the day, when people were first describing some aspects of signal transduction, they found out that there were some stimuli you could add to cells that made the cells divide. Those stimuli generated mitosis. Right? They made the cell do cell division. And so those stimuli got called mitogens. And when, again, back in the day when this is all getting discovered, people found there were mitogens, and they found that there were some transcription factors that were really important for that mitosis. And so they came up with this sort of idea that there was a transcription factor turned on by the mitogen. Turned, I know how to spell. And they made up kind of a general name for this transcription factor that was turned on by the mitogen. Okay? Does anybody happen to know what that general name is? You may have heard of it, you may not have heard of it, you may have heard of it but not get that this is where it came from. I'll start. Mitogen. Activated. Almost. Protein. So it's mitogen activated protein, or MAP. So they basically said there's got to be this transcription factor called MAP. There is actually no transcription factor called MAP. But it's like a general term, and it's going to help you a lot if you think of the transcription factor as MAP, even though that's not actually its real name. Okay? So they found this idea of this mythical transcription factor MAP. Turns out that, like with any transcription factor, MAP isn't on all the time. The cell has to turn it on when it's time to do mitosis. So most of the time the transcription factor is off. It has to get turned on. And the way that MAP gets turned on is that it gets phosphorylated. So we have to phosphorylate MAP if we're going to turn MAP on. So if you were in charge of the world, what would you name the enzyme that turns on MAP based on what I've told you so far? Yeah, Jazz. Or MAP kinase, right? You call it MAP kinase. So it turns on. I'm drawing this upside down. I usually draw it the other way around. This is feeling very like revolutionary. You have MAP kinase that is going to phosphorylate MAP and turn MAP on. It turns out MAP kinase is not on all the time in cells. The cell has to turn on MAP kinase in order to say, okay, it's time to do this whole thing. And the way MAP kinase gets turned on is that it gets phosphorylated. If you were in charge of the world and you were going to name the enzyme that turns on MAP kinase, what would you name that enzyme? Yes, Jazz.
Shot in the dark.
Okay.
MAP K kinase.
MAP K kinase or MAP kinase kinase.
Oh.
Right? MAP KK?
Of course.
Yeah, you would call it MAP kinase kinase. MAP KK. Because it's the kinase that phosphorylates MAP kinase. So you call it MAP kinase kinase. Okay? MAP kinase kinase is not on in cells all the time. It has to be activated to tell the cell, hey cell, it's time to do this process. MAP kinase kinase gets activated by phosphorylation. If you were in charge of the world, see, you're all snickering now because you're catching on to where I'm going. If you were in charge of the world, what would you name the enzyme that turns on MAP kinase kinase? Yes, Jazz.
So, like, I wouldn't personally name it.
Okay.
But I'd assume some people might call it MAP KKK.
MAP kinase kinase kinase, or MAP KKK, and that is correct. Usually people just say it out as MAP kinase kinase kinase. You usually say MAP kinase kinase kinase, honestly. You usually like say it all out with all three kinases. Okay? Make sense? All right. MAP kinase 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 is not active all the time in cells. It has to have something turn it on to activate it. The name of the thing that turns on MAP kinase 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 is RAS. RAS. You can see this pathway that I've written. The first branch after our adapter protein is a pathway that is called the RAS MAP kinase pathway. If you remember the name is the RAS MAP kinase pathway, you pretty much have all the partners. Because basically what happens is after our adapter protein is activated, we activate RAS. RAS then activates MAP kinase kinase kinase, MAP kinase kinase, MAP kinase, and the transcription factor. And so you can see all of this here. In reality, the reason why this sort of gets around Jazz's discomfort is that people don't often say MAPKKK, is because there are actually more than one protein, there's more than one protein in the cell that can serve that function. And so they all have their own individual names. MAPKKK is really the name of a group of proteins that are all serving this function, not individuals. And so the specific MAP kinase, kinase, kinase in this situation that we care a lot about is called RAF. This specific MAP kinase, kinase that we care about in this situation is called MEK. And one of the MAP kinases that we care about in this situation is called ERK. And so in reality, in immunology, immunologists very often think about RAS, then RAF, then MEK, then ERK. In other situations, there is a different MAP kinase, so it's not just ERK. Don't worry, we're getting there. It's not just ERK. It's called JNK, which is pronounced junk. That's why I said jerk, because I was like, my brain was combining junk and ERK together. So we have two different potential MAP kinases, either ERK or junk. They each, as you can see over here, can phosphorylate a transcription factor and turn on a transcription factor. ERK turns on a transcription factor called FOS. Junk turns on a transcription factor called Jun. It's actually the reason why it's junk is called Jun kinase. You can also see FOS and Jun right here. So here's RAS, RAF, MEK, ERK. We turn on FOS, we turn on Jun. And Jun is technically turned on by junk, which is the Jun kinase. Okay, do we see how all of this works? This isn't like super rocket science, right? FOS and Jun, by their powers combined, when they bind together, make a transcription factor called AP1. So that's one of our 3 transcription factors we're trying to get to. We just got there. So FOS and Jun together is AP1. You can see this here. We go through RAS and eventually get to AP1 through the MAP kinase cascade. You can see FOS and Jun coming together over here and becoming AP1. And so once we have this transcription factor, AP1, we can do some transcription. And when we were going through this whole process, one of our goals was the transcription factor AP1. So we did it. Yay, check. And so now we need to go back up to our adapters, back up to lat and SLP76, and look at another branch that is coming from lat and SLP76. So as you can see in this branch, after we had lat and SLP76 activated, we activated RAS. So we went from lat and SLP76 to RAS, as you can see here. I drew over here on this board. Another really critical protein that we're going to see after lat and SLP76 is a protein called PLC gamma. So we're also going to see the action of this protein, PLC gamma. And so you can see PLC gamma listed at the top here and see that it's our next pathway. And in order to think about PLC gamma and understand what's going on with PLC gamma, we should think about what the heck does PLC stand for, because that helps us a lot. So PLC stands for phospholipase C. So PLC gamma is also known as phospholipase C gamma. So if you just look at the name phospholipase C gamma, what does this protein do? What is the activity of this protein? Yeah, Paige. So if it phosphorylated a lipid, so Paige says phosphorylated lipid, sounds kind of right, but if it was a phospholipid, if it was that, it would actually be like lipokinase or something like that. Okay? So it's not going to phosphorylate lipids, but you're on the right track. Yeah, David. It breaks phospholipids. So we have a phospholipid, and this cuts it. This cleaves that phospholipid. You've heard about phospholipids when you've talked about membrane structures, but there are lots of different phospholipids. They have fatty acid tails. They have sort of charged head groups. And so phospholipase C gamma cleaves phospholipids. And it particularly cleaves one important phospholipid. The one important phospholipid that is getting cleaved is the one that is shown on the left here. You don't have to get super hung up on the structure. It is PI45 bisphosphate, known to most people who do any signaling work as PIP2. And so we have this molecule PIP2. You can see it's got some fatty acid tails. You can see it's got this head group. This is a structure. And phospholipase C's job is to cut PIP2. When phospholipase C cuts PIP2, there are two products made because it's basically cutting PIP2 into two pieces. One of those pieces really has the fatty acid tails, and you can see it up here. It's listed as 1,2-diacylglycerol. Again, whenever anybody talks about signaling, I can't remember the last time I said the word 1,2-diacylglycerol. That's DAG. I always call that DAG. And you can also see we sort of have this head group that is made, inositol 1,4,5 triphosphate. Again, what I always call it is IP3. So we have this phospholipid PIP2, and the enzyme PLC gamma cuts PIP2 to make these two products, DAG and IP3. So after PLC gamma has done its thing, we will have get some DAG and we'll have some IP3 made. We can also sort of think of this as another way we're having branches. We can follow what happens to the IP3. We could follow what happens to the DAG to keep going with signaling. And so we're going to follow what happens with the IP3 right now. So we're going to be thinking about what happens with IP3 first. So IP3 is going to bind to a receptor inside the cell. So there was a receptor for IP3 inside the cell. If you were in charge of the world, what is the name of this receptor? He's not here, but I still bet the rest of you can figure it out. What is the name of this receptor that's binding IP3? It is called the IP3 receptor. So IP3 is going to bind to the IP3 receptor. Okay, cool, great. The thing that's important about the IP3 receptor is sort of where it is on the cell, or where it is in the cell, and sort of some other pieces of what's going on in the cell. So the IP3 receptor primarily is found, it's this yellow thing that's shown here. I don't know why they didn't label it IP3 receptor. It's primarily found right here on the ER membrane. So IP3 receptor is chilling in the ER membrane. It's shown here as this yellow thing. Great, cool. IP3, you can see there's my IP3. It's going to bind to the IP3 receptor on the ER. The reason why this is important is that in general cell biology, the cell, the eukaryotic cell, is actually quite organized. Really likes everything in its place. Very much. I'm sure we all know people who really need everything in their exact place. And in particular, in a eukaryotic cell, all of the calcium has to be in one spot. The calcium must be in the ER. And the calcium has a charge, it's an ion. So we've got this calcium ion, Ca2+, and it's only in the ER. It's also outside of cells. The point is, it is not in the cytoplasm. The cell is like very, very, very picky. It is like, I have no calcium in my cytoplasm. No calcium ions at all in the cytoplasm. None, none, none. So you can see this situation, no blue dots in the cytoplasm. Some blue dots outside the cell. There's blue dots in the ER, but there's no blue dots in the cytoplasm. When IP3 binds to the IP3 receptor, calcium starts to leak into the cytoplasm. Calcium leaks out of the ER into the cytoplasm. And so the key thing here is that IP3 binds to the IP3 receptor, and that makes calcium fill up the cytoplasm. So the calcium comes out of the ER, goes into the cytoplasm, as you see here. So we've got this calcium release as a result of IP3. This way of drawing this is terrible. I don't know why I did it like that, but it's okay. The fact that there is now calcium in the cytoplasm where there was not before is a pretty big change for the cell. That is a pretty big sign to the cell. Hey cell, something's going on. And your cell has a bunch of proteins that bind to calcium and sense calcium. So if this change happens, if somehow calcium gets into the cytoplasm, we've got a lot of ways that we can sense that calcium. This is a key thing that's used throughout cell biology in a lot of kinds of signal transduction. You've probably seen calcium before as a second messenger, or you've heard about second messengers. Calcium release in this way is frequently a second messenger. It's part of the reason why the cell is so picky about not having calcium in the cytoplasm, so that it can use it as a second messenger when it needs to. And so if you guys have taken some certain neuro courses, like cell molecular neuro and things like that, you've probably heard about calcium signaling and proteins that are sensing calcium to transduce signals. In fact, the proteins that are involved in sensing the calcium are the same ones that are involved in neuroscience. So there are two proteins called calmodulin, and as we'll see in a second, calcineurin. So basically, if we get calcium in the cytoplasm, that is going to activate this protein called calmodulin. And so you can see calmodulin goes from being an S-shaped little worm to a different shaped little worm, and then is, as it says, active and can do something. So we turn on calmodulin. Again, why did I draw like this? This is terrible. Calmodulin. And now calmodulin can interact with other proteins. Calmodulin specifically interacts with another protein called calcineurin. You can see calcineurin here. You can see calmodulin, which looks like a folded up wrench in this picture. When there's calcium around, the wrench unfolds, and now calcineurin can do a thing. Calcineurin has a specific job. Calcineurin is a phosphatase. It removes phosphates from stuff. And in particular, calcineurin is going to interact with this protein NFAT. NFAT starts out its life with a bunch of phosphates on it. Those phosphates make it so NFAT is not allowed in the nucleus. So it's sad, it wants to go in the nucleus, but it's not allowed because of the phosphates. So here you can see NFAT. It's not in the nucleus because it's not allowed. Sad for it. When calcineurin is activated, it removes the phosphates from NFAT. So you can see that removal here. Once the phosphates are removed from NFAT, NFAT is allowed into the nucleus and it can start doing transcription. So it's a transcription factor. So you can see that the key thing here is that we're going to have calcineurin activation. That is going to give us active NFAT. NFAT is the second one of our transcription factors we wanted through this whole business. So now we got 2 of our 3 transcription factors through IP3's action. You might look at all of this and you might think to yourself, man, there's a lot of steps in this calcium business. There's a lot of stuff going on here. Couldn't she have simplified that one for us a little bit? And I will tell you that of all the parts of T-cell receptor signaling, the one that every immunologist has memorized is the calcium part. It's the one that we all really know well. And the reason why everybody knows the calcium pathway is because the first big drug that anyone invented that can influence the immune system is influencing this calcium pathway. We have this drug that actually blocks calmodulin-calcineurin interaction and makes it so NFAT can never get dephosphorylated and NFAT can never go to the nucleus. So the T cell can never be activated. It just sits around being sad and not activated. It can get its AP1, it can get its NF-kappa B, but it can never get its NFAT. And so that T cell is never active and it's sad. This drug basically makes it so T cells can't get turned on. You can see this drug on this next slide. This drug is called cyclosporin A. And so you might have heard of cyclosporin before. This is the reason why every immunologist knows the calcium pathway really well is because cyclosporin is perhaps one of the most famous drugs that impacts the immune system. And so you can see here is cyclosporin, and it gets in the middle of calmodulin and calcineurin. So NFAT always stays inactive and is always sadly in the cytoplasm. Cyclosporin A is given to everybody who gets a transplant to stop transplant rejection. Basically, transplantation wasn't really as much of a thing until we had cyclosporin to stop T cells from rejecting. Cyclosporin is also given to a number of patients with autoimmune diseases and a number of different situations. So cyclosporin was actually kind of like this wonder revolutionary drug. And it signals through this part of the pathway, which is why every immunologist knows this pathway pretty well. In fact, when I kind of first learned this whole process, I always felt a little sad because I always learned it the way I've shown it to you here, and I was like, but what about DAG? I feel bad for DAG. We just left DAG. We made DAG, PLC gamma cleaved a phospholipid to make DAG and IP3, and we did all this great stuff with IP3. And I was always like, well, what about DAG? I really was worried about DAG. So I will tell you a little bit about DAG, which is that DAG is how we get NF-kappa B. We don't have to worry about any of the steps, any of the like in-depth details of it, but DAG is the thing that leads to the NF-kappa B production. And so that's how we get our third transcription factor, NF-kappa B, in this whole process. And so this is our overview of T-cell receptor signaling. So you can see this process here, and all of the parts of it in a way that is probably much more legible than what I wrote on the board. You can see that we can get to AP1 through the RAS-MAP kinase pathway. We can get to NF-kappa B through DAG. We can get to NFAT through IP3. And thus we get transcription and gene activation. In particular, we can get transcription of IL-2, this really important gene that our T cell needs to be activated. So our goal in T cell activation was to get NFAT, AP1, and NF-kappa B so we can make IL-2, and we have succeeded in this goal. So I've got a couple little pieces regarding this that I want to talk about. So if you remember, I made this whole point about some signaling being receptor proximal signaling, and then I made this point about there also being branches. So let's think a little bit about why that matters, okay? So I told you about this drug, cyclosporin. Cyclosporin basically works right here. It's blocking calcineurin calmodulin interactions. So it's working pretty low down on this branch, right? It's not working at a receptor proximal site, it's working low down on one of these branches. So if I use cyclosporin, and I've got my 3 transcription factors, what happens to NFAT? Yeah, Julie.
I can't go to the nucleus.
Yeah, NFAT is blocked. We cannot make NFAT. What happens to AP1? Nothing. It's totally fine, right? What happens to NF-kappa B? Nothing. Totally fine. And so I'm telling you this like the only goal is you have to have the 3 combined. But actually, there's some stuff you can do with just AP1 and stuff you can do with just NF-kappa B. So here you're kind of only getting like a partial block. You're blocking this IL-2 stuff we care about, but there are some other things the T cell can still do, because you're blocking something further down on a branch. Now let's imagine I have a drug that blocks LCK, or a drug that blocks ZAP70. So I'm going to block LCK or ZAP70 up here. Now what's going to happen? Yeah, Jazz.
These 2 are both AP1 and NF-kappa as well.
So now you've lost all 3 transcription factors. So when you see events that are happening in those early receptor proximal steps of signaling, you influence multiple pathways, you influence multiple transcription factors. While if you try to sort of do things at later steps, you might only impact one pathway, one transcription factor. That becomes really important for us as immunologists when we're thinking about how to treat different diseases, because we can think about, do we want to block them all? Or is that like going to give us severe side effects and maybe we only want to block one? And so that sort of, do you block at the top, do you block at the bottom, is actually a huge deal with all of this. So I've got about 10 minutes left. I've got some stuff here to talk about with regard to IL-2. But before I tell you about IL-2, we'll have a brief interlude. So if you recall, I told you previously that the person who I TA'd for, Shiv Pillai, wrote immunology poems, or raps. I showed you guys a lymphocyte rap. about different aspects of things we have talked about this semester. And he actually wrote a poem about all of this, or wrote a little story about it. Weirdly enough, this one, the YouTube video doesn't work. It got cut off. And so I emailed him and he sent me the text. And so I am going to recite it for you. I am not nearly as good at it as he is. And so basically, what you're going to hear in this is me going through this pathway for you. One thing I will just tell you, at a couple places it mentions YXXL. That is the amino acid sequence in an ITAM. That's the tyrosine in the ITAM, okay? And as you're hearing this, you should be thinking about like the Iliad and Odysseus and the Odyssey, because it's a Greek epic. O Hercules and Odysseus, great heroes of the past, we lymphocytes are forsaken. We fear we may not last. This next part is a spoiler for next week. Like you, we're xenophobic, and at our very core, political correctness wholeheartedly deplore. You Greeks had mighty heroes, each like a lymphoid clone. They defended the homeland, faced danger all alone. Came a foreign invader, and an army would be raised. 'Twas clonal expansion, for this Zeus be praised. No room for any mercy, that is our battle cry. Self we tolerate gladly, non-self must surely die. We always had our enemies, but now face gross abuse. Lord, our signals go unanswered. We've got the cyclosporin blues. Again, spoiler for next week. You see a Phoenician trader, you know it bodes no harm. For a dreaded Persian warship, you must sound the alarm. Selective xenophobia, no B7 means you're spared. Second signal spells out danger. For battle, we're prepared. The TCR sees its peptide in an MHC array. Co-receptors bind MHC, bring LCK in play. Those YXXL tyrosines now have phosphates to show. ZAP70 hugs an ITAM, and the cell is good to go. No room for any mercy, that is the battle cry. Self we tolerate gladly, non-self must surely die. We always had our enemies, but now face gross abuse. Lord, our signals have gone crazy, got the cyclosporin blues. ZAP70 gives PLC a very gentle shove. PLC and LAT are now very much in love. The enzyme releases IP3. It floats into the cell. On its ligand-gated channel, this sugar casts a spell. From ER and mitochondria, calcium begins to flow. The cell is soon suffused with a calmodulin glow. With calcium calmodulin, calcineurin forms a trio. They samba together like it's carnival in Rio. No room for any mercy, that is our battle cry. Self we tolerate gladly, but non-self must surely die. We always had our enemies, but now face gross abuse. Oh Lord, we're going crazy, got the cyclosporin blues. Next part I didn't actually tell you. The early calcium efflux depletes the ER store. Calcineurin, like Oliver, asks the cell for more. Stim1 links the ER to the wavy cell membrane, tags ORI the crack channel to join the calcium game. From the surface of NFAT, a phosphate group is lost. No cytosolic retention, the nuclear pore is crossed. The promoters then awaken. Cytokines begin to churn. From G1S to M phase, the cell will now turn. No room for any mercy, that is our battle cry. Self we tolerate gladly, but non-self must surely die. We've always had our enemies, but now face gross abuse. Oh Lord, we're going crazy, got the cyclosporin blues. My host has moribund kidneys. Bad kidneys. Have we not fought with honor? Must we now be deceived? The TCR is activated. A calcium high ensues, but calcineurin lies frozen. We got the cyclosporin blues. See, you got a new kidney transplant? NFAT lies frustrated. Promoters fail to churn. The engine keeps on cranking, but the wheels refuse to turn. Once we fought with honor, but now in hell we burn. Consecrate our ashes in a drug-drenched hero's urn. I know from the past that one was hard to pronounce. No room for any mercy, that was our battle cry. Pathogens we dealt with gladly, but it is now our turn to die. We vanquished all our enemies, now we must learn to lose. Open those gates to Hades, we've lived the Cyclosporin blues. The end. So hopefully that was a helpful way for you to think through this process. And the goal of this process was to make IL-2. So now we're able to transcribe IL-2 and make the IL-2 protein. And so the next thing that we kind of have to think about is, well, then what the heck is IL-2? And who cares that we make the protein? We make the IL-2 protein. You might guess from the name that IL-2 is, of course, a cytokine protein. And that might start to ring an alarm bell for you. Because remember that one of the things I told you when we think about activation of a T cell is that there are some important cytokine signals that have to happen. And so this is why I kind of jump ahead to signal 3, because signal 3 can come from IL-2. And so basically the T cell is going to potentially start making IL-2 to help contribute to this signal 3. When IL-2 was first described, IL-2 did not have the name IL-2. This is before we had the name IL-2. And the name that this cytokine had before it got the name IL-2 was T cell growth factor, TCGF. And so you can, from that, imagine what IL-2 does. So what do you think IL-2 does? Yeah? It induces proliferation of T cells. It also induces survival of T cells. So it both makes the cell go into cell cycle, which you can see over here, as well as increasing survival of that T cell. And so in order to have, and it really also will help our T cell get activated. It will help our T cell go from naive to turned on, activated all the way T cell. And so you can see this process happening here. You can see our cells activated. It does some divisions, makes a bunch of progeny. Those progeny are different because they have these letters in them that they didn't have before. So these cells have been activated. And what you can also notice is that the T cell can make IL-2 and basically feed it to itself. It makes cytokine that it then gives itself. And so that T cell can make IL-2 and then signal that, which will signal back to itself to induce proliferation and activation. That is known as an autocrine process of IL-2 signaling. So basically what we can see in this situation is our T cell, once we start this activation process, it starts making IL-2 and feeds itself that IL-2 to further activate it in a process that's autocrine. Oftentimes what we see, it's not shown so well on this image, but on other images, I know from other parts of the textbook it is shown. Oftentimes you'll see multiple T cells kind of interacting with this one dendritic cell or this one antigen presenting cell. So imagine maybe there are like 5 T cells doing this whole thing right here. They also could feed each other IL-2. So some of the IL-2 is autocrine, some of the IL-2 is paracrine, where they're feeding their neighbors. But it's not like there's IL-2 going through the whole body. It's just there's a little bit of IL-2 right in that location so that all of these T cells can sort of feed each other towards activation. I've got a few more details to tell you about IL-2 and how some of the details of IL-2 signaling works. And then we're going to go into signal 2 on Monday, as well as some of the next parts of T cell effector function. So please make sure you reply to the doodle poll by Monday's class, and also remember the homework due today by 5.